Myers is making is it's overwhelmingly pointing towards a creator. And I think as you go through some of this, you will see that as well. First week, he looked at creation from a scientific perspective. Where did the universe come from? And just how vast is it? And all the stuff that's tied with that. From the second week, he looked at it from a physics perspective and how finely tuned the universe really is and the odds behind that just happening, which are astronomical, if you don't mind the pun, but it's just crazy to think that there wasn't some kind of designer behind it. And again, the evidence points to a single beginning, just like we see in the book of Genesis. We might not have all our facts together yet, but what you see points to a single beginning, and the tuning of the physical properties of the universe points to some form of a designer as well, which again would tend itself towards a supreme being. You know, when we looked at some of that, I think about God speaking to Moses. And Moses asked him, "What should I tell the people is your name?" And he says that I am that I am. And I've often puzzled over that response. But if God is a being that is outside of the universe, which a lot of the evidence points to, then how could you describe yourself? How could God describe himself in a way that we would understand? And I've often looked at that and thought, you know, why would he say that? But I'm starting to understand, you know, if you come from outside this universe and created everything, there's no way we could understand all of that. So he just used the name that best fit. But anyways, this week, we're not going to look at the universe. We're going to go small and look into a living cell and look at evidence in biology for design. Not until just recently, within, I'm going to say, the last 20 years or so, have we been able to see what's inside of a cell, and for the longest time they just thought it was a mass of protoplasm. And they're finding out there's a lot more going on inside the cell than we ever thought. So what did they find when they got in there? And I'm going to tell you right up front, you're going to get presented with a lot of information in this video that you might find rather confusing. But what I want you to focus on, if you don't get the gist of everything, is just how complicated it is and what the possibilities are of all of this just happening. And we'll discuss it more in detail when we're done here if we've got time. This is about a 30-minute video, but you'll have to pay attention or you're going to get lost. And we'll get started into it now. Get back here.
Remember when we were talking about Richard Dawkins? Remember he used that example of the garden and how if you look at a beautifully planted and arranged garden, it's natural to think there must be a gardener. But Charles Darwin explained that there is no gardener behind the garden. And he's using the garden as a metaphor for all the appearance of design in living things. The new atheism is built on this idea that there's no evidence of design in nature, that the design is an illusion. And we're going to see that today as we start talking about the debate about design in biology, that there's two different sides. There's the perspective that things look designed because they really are designed. That's the idea of intelligent design. And then there's the broadly speaking, the Darwinian point of view, which says things look designed, but they're not really designed. And since they're not really designed, then we have a worldview implication. And that is that there is no God, or as Dawkins said, it's very, very unlikely that there is a God, right? So the question of design, which we've already addressed when we talked about the fine-tuning in physics and cosmology, is a critical worldview shaping question. If you accept that there's design in physics, that seems to point to some kind of designer beyond the universe as a whole. But now we're going to come to the area of biology, where the conventional wisdom, at least for the last 150 years, is that there's no design, that there's an appearance of design, but things that look designed are actually not designed. That appearance is an illusion. That's the classical Darwinian point of view. That's Dawkins's point of view, and that's why he's so confident that God is so very, very, very unlikely, okay? So let's dive in and look at this question of design in a little more detail. Okay, here's Dawkins. He says that biology is the study of complicated things that give the appearance of having been designed for a purpose. What's the key word there from the Darwinian point of view? Appearance, right? The appearance is an illusion. It's not real. They think that there's a scientific explanation for the appearance of design that does not involve any actual design, no intelligence, okay? And the scientific explanation or mechanism that they propose is natural selection. Natural selection is an undirected mechanism, according to Charles Darwin, that can mimic the powers of a designing intelligence. Natural selection selects for beneficial variations, or in our modern times we'd say mutations, and incrementally it produces these complex structures with no guiding hand whatsoever. It's entirely an undirected mechanism. So Francisco Ayala, the past president of the American Association for the Advancement of Science, has put it this way. He says that the functional design of organisms and their features would seem to argue for the existence of a designer. It would seem to. But it was Darwin's greatest accomplishment to show that the directive organization, the complexity of living things, can be explained as the result of a natural process without any need to resort to a creator or any other external agent. No guiding hand, no design, no creator, no need of that hypothesis. So the naturalistic worldview is a consequence of this way of thinking. If there's no designer, God talk then becomes delusional. And that's the title of Dawkins' book. Okay, what we're going to do today, we're going to ask ourselves whether or not there is any evidence of actual design in nature. There's two parts of evolutionary theory. There's biological evolution, which explains how you get from simple, pre-existing forms of life to more complex forms of life. And you may remember that Darwin used a branching tree diagram to illustrate his ideas about the history of life. All the branches up here represent the forms of life that exist today. The giraffes and the crocodiles and the fishes and the birds and human beings and the bacteria. Everything that exists is on one of these branches. But the idea of Darwin's theory was that if you go back far enough, you get to a single common ancestor from which all these other things descended or evolved. What we're going to look at today is another part of the big evolutionary synthesis that's called chemical evolution. And that's how you get to the first cell itself. Because that turns out to be a pretty important problem. How do you get from simple non-living chemicals down here in the soup to the first one-celled organism? We're going to ask ourselves the question as to whether or not there is any appearance of design at the level of that first cell, and whether, if so, whether that appearance of design can be explained away by some undirected mechanism, or whether it's better explained by actual intelligent design. Let's talk about the simple cell. If you go back to the 1860s and 70s, in the immediate decades after the publication of The Origin of Species, scientists decided to extend Darwin's way of thinking back further in time. Darwin starts here and tries to explain how all the other branches on the tree arise. But he doesn't attempt to explain how you get the first life. Scientists in the immediate years after that tried to do that. They came up with a theory of chemical evolution, chemicals to first life. And they didn't really think it was much of a problem. They didn't think it was a hard thing to do. They weren't really impressed that there was any appearance of design that needed to be explained, because they thought the cell was a simple, homogeneous globule of plasm. Okay? Like red Jell-O on your Christmas plate or something. You know, that's what the cell's made of, something real simple and gooey. And if you have that concept of the cell, it's pretty easy to see how in one or two steps you might be able to have some chemicals combine and recombine, a few simple reactions, and voila, you have the stuff out of which life is made. They call that stuff protoplasm. And Thomas Henry Huxley tried to explain it, the origin of that first cell, as a result of a few simple chemical reactions. Well, by the 1890s and then into the turn of the new century, scientists were learning more and more all the time about proteins. And they soon learned that proteins were really big, long, complicated molecules, and that there were metabolic reactions, interactions between proteins and other molecules in the cell that were going on. And as we got an idea about metabolism and all the complexity of the chemical reactions that are going on inside cells, scientists realized that this one or two step chemical reaction explanation for the origin of life wasn't going to do. So another scientist came along, and he was a Russian, Alexander Ivanovich Oparin, okay? And he came along in the 1920s, 1930s, and proposed a more credible chemical evolutionary theory that had not just one or two steps, but a whole bunch of steps. And his idea was if you start with a bunch of atmospheric gases and you zap them with some kind of energy source, either ultraviolet radiation or lightning, then you're going to get some reactions and simple building blocks will form. Building blocks of larger molecules. And we're going to think of the building blocks today like these snap-lock beads. Do you guys remember these? Anyway, so Oparin proposed that if you had the right chemicals in the atmosphere and they interacted the right way, you could get amino acids to form through a series of chemical reactions. Then the idea would be the building blocks would link up by other chemical reactions and form these long chain-like molecules, which we call today proteins. Proteins are made of a string of amino acids that are arranged in a precise way. Then those molecules would interact with other molecules, and eventually you'd get metabolic reactions going on. At some point, some kind of enclosure with some membrane-like thing would surround all these chemical reactions, and then you get a competition for survival among the little membrane-like things, and the ones that were better at taking in nutrients in the competition for survival would survive, the other ones would die off. As a result, they get more and more complex, and at the end of that long process, you'd have the cell that we know today. Anyone encountered this theory? Have you ever heard of this? Run into it in your biology textbooks? It's still taught today in many of them. You probably have encountered this experimental apparatus, because this was an experiment that was trying to test one of the steps in that long process of chemical evolution from simple chemicals to the single cell. What Miller did was he did what Oparin imagined. He put some gases in a vessel, sparked them, gave them some energy, allowed the byproducts to collect in this trap down here. And after a couple days, he was able to detect 2 or 3 of the amino acids that form proteins. And this was performed in 1953 and heralded in the press and in the scientific world as a great breakthrough in understanding the origin of life. And let's see why. If we go back to the previous slide, what Miller was able to do was simulate getting from here, the simple gases in the atmosphere, to here, the building blocks, the amino acids, in what was called the prebiotic soup. This was an imaginary idea that people had about what the early Earth was like, a great big ocean full of amino acids that could interact with each other. Can you see any problems, though, with this experiment?
Where did you get the gas from?
Interesting. That turned out to be a big problem. As people have learned more about the atmosphere, those were not the gases that were likely present. In fact, the gases that were present were more neutral or even slightly oxidizing, which means that you would not get spontaneous formation of amino acids in those kinds of conditions. The other problem, though, is much more significant. And that has to do with this step right here, okay? Getting from here to here. Because it turns out that proteins are very interesting molecules. They have some very important properties. And one of them is that in order for them to do their jobs, you have to have the amino acids lined up in a very specific and particular way. You can't just arrange them in any old way and hope that the proteins are going to do what you want them to do. If you get the amino acids to line up in the right way, then they exert forces on each other, and those forces cause a chain to fold up into a specific shape, okay? And that shape enables the protein to do a very specific job. It's got a hand-in-glove fit, all right? And what Miller was able to do was to get from this step here to here, but he didn't really have any idea how you would get those amino acids to line up correctly. One of my graduate school supervisors back in England studied the origin of life. She was a specialist on the history of research about the origin of life, and she made an observation that was really interesting. She said, at the heart of the problem of the origin of life lies a fundamental question. It's a pretty straightforward, obvious question. What is it we're trying to explain the origin of? So the first thing we need to do when we're talking about the origin of life is begin to think about what exactly it is that has to be explained. If we're going to explain the origin of life, well, let's study carefully what's in life so we know what we've got to account for. Proteins are absolutely essential for what goes on inside the living system. Think of it like a toolbox. If you've got a toolbox, you've got a hammer, a saw, a plane, a screwdriver, a little hand drill maybe. Each one of those tools performs a function in virtue of its shape. The shape and the function are related. You can't nail a nail with the saw very effectively. It's just not got the right shape or consistency. It's the same thing with proteins. They do hundreds of different jobs in the cells. They're structural parts for motors within the cell. They catalyze reactions within the cell. They're enzymes. They also process information on the DNA strand. And every one of the jobs that a protein does depends on the specific shape of that protein. So one of the first things that people discovered about proteins is that they have a very complex three-dimensional shape. And here's some representations of protein shapes. There's hundreds of different proteins. Here we've got a protein that catalyzes the breaking apart of a two-part sugar, a disaccharide. Notice, here's the sugar, and here's the little pocket in the enzyme that catalyzes the reaction. Notice the fit there. Notice how tight that fit is. That's the way proteins work. So that's something, if you're going to explain proteins, you've got to explain that complex three-dimensional structure and that complex three-dimensional shape and the specificity. We have a pretty good idea of how that arises now. Remember I was saying how the amino acids have to be linked together in a particular way, and if they're linked together just right way, you get the thing to fold right. If you rearrange them willy-nilly, you may lose all that beautiful balance of the forces, and the thing may just go limp, okay, and not perform any function at all. So what we say about proteins is they have a property called sequence specificity. And if something is sequence specific, that means that the function of the whole depends upon the arrangement of the parts. Can you think of anything else that has sequence specificity? Those of you who are taking notes?
Words?
Words? Okay, absolutely, absolutely. Grammar, sentences, words. This is one of the things that Francis Crick first observed, that proteins are very much like words, that you have an amino acid alphabet, and depending on how those amino acids are arranged, you're either going to get a functional protein or maybe something that's just garbage floating around, okay? So you can draw an analogy to language. If we arrange the letters correctly, then they're going to perform a communication function. If you arrange the amino acids correctly, you're going to get a protein function. So function of the whole depends on the sequence of the parts. So proteins, got to explain them. They got three key features. The shapes of the proteins perform the functions, the folded chains form the shapes, and then the precise amino acid sequencing is responsible for both the folded chains and the functions that are performed. So if you're trying to explain the origin of life, what do you think is the most fundamental question then? What's the thing that really is responsible for the protein function?
The order?
The sequence, the order, right. So where does that come from? All right. Well, it turns out that we know something about that now. Okay, we know there's another molecule in the cell called DNA that encodes instructions, interestingly. It's a machine code or digital code along the spine of the DNA molecule that actually directs the cell, tells it how to build or how to arrange those amino acids. Now, this idea was first proposed by Francis Crick in 1957, a few years after he and Watson discovered the double helix structure of the DNA. Crick had this fantastic hypothesis a few years later. He said, gee whiz, I think the arrangement of the characters along the spine, there's chemicals on the inside. Let's see the actual chemical structure of DNA. You've got a sugar and phosphate backbone, and then on the inside of the molecule, you have these chemicals called bases. And this was Crick's idea, that the bases on the inside of the molecule function just like alphabetic characters in a written language, or digital characters like zeros and ones in computer code. And that they direct the synthesis of proteins within the cell. So we think about how do proteins arise with their beautiful shapes and their perfectly arranged amino acids? Well, they arise that way because in the cell there's another molecule that has instructions that direct the cell and its machinery to build the proteins in just the right way. Okay? So now we're kind of pushing the mystery back a little further because now we've got another question. And the question is, what? Yeah.
Who put the message inside of the DNA?
Yeah, exactly. So that's something we're going to be looking at real closely. In the meantime, I want us to get a real clear picture of this sequence hypothesis of Crick's because it was confirmed by a series of experiments over about 5 years after 1957. And it's basically his idea was the arrangement of characters in the DNA is responsible for telling the cell how to construct the proteins. We have something very similar to that up in Seattle, where I'm from. We have a plant at Boeing, and they use a technology called CAD-CAM, Computer Assisted Design and Manufacture. And you all are probably pretty familiar with this way of doing things because we all live now in a world of digital computing. But what they do at Boeing is they have a machine with all these digital instructions. They're translated into another kind of code, which then directs a machine to build other machinery, like a machine that would place the rivets in a particular place on the wings of an airplane. All that's controlled by digital code. That's what Crick was proposing about the cell, and that's what we now know to be true about it. Let's look at a little piece of animation that shows exactly how this plays out. What we're going to see is how the information in DNA directs the synthesis or construction of proteins.
In 1957, Francis Crick first proposed that chemicals, called bases along the spine of the DNA molecule, function as alphabetic characters in a written language or digital characters in machine code. This short animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule being constructed inside the polymerase. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation ensues. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cells by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acid determines the type of protein constructed. When the construction of the chain is complete, it is often transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
We have cells, the smallest unit of life. We've got trillions of them in our bodies, and these processes are taking place at phenomenally rapid rates every second, right now as we speak. Okay? The transcribing or copying of the genetic message, the translation of it into a protein, and then the folding of the protein so it can go do its job. Yeah, Nathan?
Do we know what guides the movement of all those pieces?
We do have some ideas. Some parts of the animation, even though they look really complex, they vastly oversimplify it. That little strand of RNA does not just magically know to go there. It's carried along by other proteins and other parts of the cells that have a specificity, and they guide it there. So everything in the cell runs by the principle of specificity, hand and glove or lock and key fit. Very exciting. Okay, now, so anyway, the big point here is that information is the key. Information directs the show within the cell. The information in DNA is what's directing the machinery to construct those proteins in the right way. Now, I want to offer just a quick clarification about what kind of information we're talking about. If any of you have encountered the idea of information in a math class at university, you're going to encounter the idea of Shannon's mathematical theory of information. And Shannon had a great intuition. He thought that information and probability were inversely related. And here's how he got to this idea. He said, information intuitively is about the reduction of uncertainty. So if I tell you that it's going to rain in November in Seattle, have I told you, at least if you're from Seattle, anything that's terribly informative? Not very informative statement. It doesn't reduce much uncertainty. I know it will rain in November in Seattle, usually almost every day. If I tell you, however, say in 1960, that on May 18th, 1980, Mount St. Helens is going to blow up, that's pretty specific information, because that's exactly what happened, okay? So that would reduce a lot of uncertainty, all right? So what Shannon did, the great mathematician, is he linked this mathematically to the idea of improbability. But this is what I want you to zero in on. I could pick out Scrabble letters out of a bag right now and get something very improbable, but does it convey meaningful or functional information? Okay. According to the mathematicians, it's an improbable sequence, therefore it has a measure of information. But it's not the kind of information that we are used to talking about, where the information performs a function, where it conveys a meaning. It's important to understand what we're talking about in biology. The information in DNA, what we'd like to say is it's not just complex, which is a synonym for improbable, it's specified and complex. So this top sequence has Shannon information. It's complex, it's improbable, but no meaning, no function. What about this bottom one? Can you see that there's a difference? Time and tide and wait for no man. That's a meaningful or functional string of characters, right? Now, what do we have in DNA? We have specified information. We have information of this kind, not this kind. The information in DNA performs a function. What's the function it performs?
Creating proteins.
It tells the cell how to arrange the amino acids to build the proteins, right? So, and Crick was clear about this right from the very beginning of the molecular biological revolution, where people are discovering all about DNA. He says, by information, I mean the specific arrangement of the amino acid sequence. Information here means the precise determination of sequence, either of bases in the DNA or of amino acids in the proteins. So he says, the information in biology is not just Shannon information, it's specified information. It's information that performs an important function. Okay? Now that's really clear, because remember my supervisor's statement. We're going to explain the origin of life. We've got to be clear about what it is we're trying to explain. What's the fundamental mystery in life? It's the origin of information. What kind of information are we talking about? We're talking about specified information, functional information, not merely an improbable arrangement of characters. And the leading scientists in the field of origin of life biology have been very clear about this. German biochemist Bernd Olaf Küppers says that the problem of the origin of life is basically equivalent to the problem of the origin of biological information. Okay? That's what needs to be explained. But let's go back to Richard Dawkins, where we started the lecture. Remember, he was saying that when we're looking at life, we're looking at something that appears to be designed, but isn't really designed. And when people took that idea and began to try to use evolutionary concept to explain the origin of life in the first place, they were really confident that they could do it because they didn't think there was even an appearance of design. The cell was thought to be so simple, they thought we didn't even have an appearance of design. Now we have an exquisite appearance of design. What we're talking about is digital code inside the cell. And Dawkins, oddly, has been one of the people who's acknowledged that this is a big mystery, but also that the information in DNA is, he says, like a machine code. It's uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be interchanged with those of a computer engineering journal. In other words, we've got a striking appearance of design here. Now, the question I want to leave you with at the end of this lecture, and we're going to come back to address in the next lecture, is this. Has this appearance of design been explained away by any undirected process? Remember, the whole Darwinian strategy at a higher level in biology is to explain away the appearance of design using the undirected process of natural selection acting on random mutations and variations. Can that strategy work when we're talking about the origin of life? And in particular, the origin of the information you need to get life going? If so, then the conclusion of no design, which is driving the confidence that Dawkins has in his atheistic and materialistic worldview, might still hold. But if not, if we're looking at evidence of real design, we may be looking at a new day in biology, and one which is going to require an entirely different perspective.
Now, I don't know about you, but my head sort of spins after looking at all that material. Just remember this. The only source for specified functional information is an intelligent source. Now, the reason that's important is because you should know that you don't have to be bullied or intimidated in the biology classroom about your conviction about God.
Okay. So some things to talk about today. I don't know about you all, but I find this stuff highly interesting. And a little
We're real
beyond me,
familiar
to
with
be
DNA.
quite honest.
Where did that
I
information come
understand
from?
it
This
only
time
to
we're
a
going to look
point,
at the
but after
competing
that,
explanations,
I am no chemist by
prebiotic, natural
any
selection,
way, shape, or
self-organization.
form,
Let's
but
try
I
this
do
chance
know
idea.
chemistry
How
plays a
likely
big
is it that we'd ever
part
get
of
a
our
protein
life,
to arise
how
by
things
chance?
work in this world.
Would you mind just writing down whatever letter you
Couple
got
things to
out of the
talk
bag?
about. So he
Let's see if we
discussed
can,
about
by
the theory of
chance
evolution
alone,
in two
come up
different
with
parts.
some specified
The first
information.
one is the biological
No serious
evolution,
scientist thinks this is the way it
which
happens.
basically it was
I know
Darwin's
about the odds, and
theory that we
eventually
started
the
from a
gibberish is going to
single-celled
completely
organism
swamp
and
the
became
swamp,
who
that
we
is,
are today simply by
the
selective
any
process.
meaningful sequence.
I've always had
This is
hard
not a good
time with
hypothesis,
that. I can
okay?
see some
Something
sense
else
in
is
it,
at work.
But when I start adding all this other stuff up,
You're not
it
just
don't
dealing with
add up
chance,
anymore.
so don't get bullied on this one.
The other piece he talked about was chemical evolution. How you take simple chemicals and form into them something that produces life naturally without any design intent whatsoever or any outside forces working on it. That's the part that puzzles me. And he talked about the arrangement of amino acids in a very specific sequence, like that chain of connective balls that we probably all played with when we were kids, how each one of those balls has to be in the right place, or this protein that it produces does not perform any specific function. And I liked what he had to lay out there about our own language. We have an alphabet, and if you don't arrange those letters in a specific order, you get nothing functional out of it. And then the natural selection of creating even a single word that means something just doesn't happen, let alone a whole sentence of words with spaces in the right place and all that other stuff. So I like that analogy, the comparison that he does with that. It's very much similar. I can't help but think about who we are and the trillions of cells that's within each one of us. And we think about function. You know, he talked about the arrangement of amino acids to form a single protein and how that works. We are trillions of cells with all of that stuff going on inside, and the thing that really sets my mind afloat is all of that came from the information that existed by the joining of two cells. Each one of us is a product of that in our own unique way. And what separates human beings from raccoons or anything else, all of that information is in the joining of two cells. I don't know how in the world, and we perform a very specific function. Each part of our body has its own individual function, just like a protein does in a cell. And if you don't arrange that stuff right, it don't work. So I don't know how you get around the idea of there being no design. I don't see it. It's not plausible for me at all. Questions? Ideas? Yes, Roy. all of us got to have a foundation for our faith. And our faith is nobody's faith but our own. I can't put faith into Russell. We can have discussions and everything else. It might help build each other's faith, but in the end, Russell's faith is his own, and my faith is my own, and your faith is your own. And the more evidence of God that I find, the stronger my faith becomes, and I reach a point where I no longer question whether God exists or not. I don't understand everything. I don't fully comprehend everything. But the evidence tells me that there is a God, whether I understand it or not. And if I get to that point, then all the other stuff that's in the Bible that I don't fully understand becomes plausible to me. The existence of God becomes plausible, and therefore my foundation of faith becomes stronger. And the things I don't have answers for, I set aside until I do. My belief still stands. Craig.
I was just going to say, I think if I was trying to help explain to someone or to maybe try to convince them to see the light about the existence of God, I would definitely, what he explained today seems to be a lot more convincing, because when we talk about the big picture, the astrological explanation, they get away with using these highly theoretical things so they can just explain, okay, everything came from a single point. But with the cellular level, we're talking about information, and to me that's just a much more convincing argument to have to explain away how that information came. So I really appreciate that video we saw today.
Yeah, he'll continue on to that. I haven't decided yet whether I want to show you the next lesson in this series, because he goes even into further depth on how this code that's within the cell is more like the alphabet than we know. It's very specific in how it's arranged, and if it's not arranged that way, it doesn't function worth a hoot. And I continue to think about this. He mentioned Boeing, and of course you guys know I build airplanes for a living. And I know how hard it is to take raw materials and assemble 30,000 parts, which is nothing like the trillion parts that's in our or trillion cells that's in our body. How to take 30,000 parts and make them into a functional aircraft and have it function the way we intended it to. That don't come easy. There's a lot of trouble along the way. Yet you and I are sitting here with trillions of cells, each one of it with its own function, and all of that came from the information in 2 cells. Remarkable stuff. Anyways, we're running over time. I'm going to share a prayer, and then we'll dismiss for a little while and get into our worship services. Our Heavenly Father, Lord, thank you for providing us evidence of your existence. Lord, this is some remarkable stuff to see. And knowing how all of this works, Lord, I just can't help but think how great you truly are. You have understanding of stuff that is way beyond our comprehension, Lord, and coming from where we used to be thousands of years ago when Jesus walked the earth, and our understanding of how it works to where we are today, all that does is just show us more and more that you do exist, Lord, and I'm of the opinion we have just scratched the surface on what you've really created. So, Lord, thank you for what we've got to witness today, Lord, and may we all go home with a little bit stronger measure of faith than we had when we came. It's in Jesus' name we pray. Amen.